All right. Well, it's a real pleasure for me to introduce Becca back, who's a third year graduate student. Okay, fourth. In the genetics program here, Becca has sort of single-handedly advanced a brand new direction of research in my lab, which is using droplet microfluidics to study biology. I'd say the level of independence and maturity and drive that she's shown has been really quite exceptional. And so the work she's about to present on studying bacteriophage-host interactions in droplets is almost entirely her own brainchild. So, kudos to her for that. I'll maybe say one quick thing before I hand the stage over to her. So in addition to being a droplet expert with bacteriophages, it turns out that Becca is also an expert at 3D printing bacteriophages. So she's printed all kinds of 3D printed toys of phages which look like pencil boxes, lock boxes, maybe a beer mug.
Yeah.
And so so these, again, these 3D printed phages have been a huge hit with the kids at the Wisconsin Science Festival, where she's participated annually for the last couple of years. And it's really amazing science outreach. And so it's a pleasure to introduce her. Thank you.
Share your screen here on Zoom.
Hi, everyone. Thank you, Watson, for that introduction. My name is Becca, and essentially I am a now fourth-year genetics PhD student, and I'm really excited to talk about my work, which is massively parallel single-cell sequencing to study phage-host interactions. So as a brief intro, I decided to make my table of contents slide basically as a roadmap. Since I mostly work on technology development, you can see there are a little bits of highs and lows, the peaks and valleys of developing a new tool that you can use. So I'll be doing a brief introduction and background of what I'm exactly studying, and then introducing my method and all the validation steps that I have been working on the past couple of years, as well as my me addressing the RCR that the NIH wants us to do. So phage-host interactions are ubiquitous and abundant. So bacteriophages or phages are essentially viruses that specifically infect and replicate inside bacterial hosts, or as I'm going to call them, hosts. So these kinds of interactions mediate all different kinds of different microbiomes, such as the ocean microbiome, the soil microbiome, and the gut microbiome. So they actually mediate the composition and diversity, and you can see that there are pretty high abundances. So in the ocean microbiome, you can see right here, there's at least 5 times 10 to the 12 different kinds of phage-host interactions present per square meter of water. If you look at soil, they're also there at least 10 to the third different kind of interactions per gram of soil. And for the gut microbiome, there's about 10 times 8 at least within a gram of feces. So because of these interactions and how ubiquitous and natural they are, there's actually a revitalization about using these phages as a microbial editing tool to treat microbial dysbiosis. An issue with this, however, is that there are many, many different kinds of strains of bacteria. So if you can imagine, there are thousands of phages times 300 to 500 different kinds of strains. You could just imagine the infinite number of possibilities of host-phage interactions that are too complex to elucidate. So phages in general can be very general or unique to certain kind of bacterial strains. So you can see in these lines right here, this means that a phage can be able to directly infect and lyse the host. Or right here in this line, it shows that there could be some resistance or some sort of fitness advantage that the phage can have to actually infect this kind of bacteria. However, this gets a little bit more complex when this is a heterogeneous pool of different kinds of phages and bacteria. So you might have resistance emerging for different kinds of bacteria, which is defense mechanisms, or there might be phages that also accumulate point mutations that might be able to allow them to predate on different kinds of bacterial strains if they never were there before. But the real question that we're trying to seek, that all phage biologists are trying to address, is who is interacting with whom. And right now, we do not have the tools to do that. So there are some tools that actually can address studying different kind of phage-host interactions, and these are traditional bulk assays. So this could be within tubes or OD 600 plates or plaque assays as we use for virologists. But these only test one by one, and it's difficult to elucidate basically the kind of heterogeneity, whether it's genetic or phenotypic, within these kind of flasks. And these methods also cannot scale to actually capture the complete complexity of all these different interactions. As Watson introduced before, I study droplet microfluidics, and the reason why we actually want to use this tool is because all contents are contained within each droplet, and this could also separate all the different kind of complex heterogeneous samples into single cells within each droplet. But as you can see, the space for different kind of ultra high throughput methods is very empty, so there's a lot of potential to expand on this. So droplet microfluidics is essentially the manipulation of fluids. So over here, I have a standard microfluidic device. This is the oil right here that could pinch off whatever aqueous solution that you want. And then you could collect millions and millions of droplets right here. And it's highly customizable. These are some of the microfluidic devices that I actually designed in lab. And this is just the video showing. Never mind. This was supposed to be a video. A video showing how the droplets are basically generated and pinched off by the oil. So if you're going to ask me why droplet microfluidics, I would say because you could save costs and because you could use very limited amount of reagents. But if you're to ask me at a deeper level, I would say that the advantages of using droplet microfluidics is very vast. So you could actually confine samples within ultra small volumes. So you could have a milliliter of a sample and you could actually get picoliters, millions and millions of different reactions. So you could actually be able to study the different kinds of screenings within these millions of droplets. In addition, it could handle incorporation of complex liquid handling. So this here is a video where you could actually inject reagents. So if you want to add a reagent that you kind of don't want in the reaction mix before, you could actually add it in the middle of your experiment during droplet microfluidics. In addition, the type of droplets that you could be using is actually very tunable. So for example, in tissue engineering or for studying different kind of drug release, you could actually use different kinds of hydrogels to basically compose the droplets into different kinds of things. So you could actually culture cells within, let's say, like agarose or collagen. And also, microfluidics is a multidisciplinary space. It's not purely for engineering. Other different kinds of fields have been using it to study things such as C. elegans and even plants. So it's very applicable to various research fields. So basically, there's a novel research opportunity that stems from this lack of high throughput methods. So what we seek to do is to actually develop a tool that could demystify all these different kind of phage-host interactions in complex communities. So some things that we wanted to consider when developing a tool is, can it actually replicate the natural biology that goes on in bulk conditions? Can we compartmentalize each of these individual pairwise interactions? And because of droplet microfluidics, we want to basically develop a high throughput and massively parallel tool that could help us identify different kinds of heterogeneity that's going on. And in general, our ultimate goal is to make a tool that is generalizable to various different kinds of microbiologists. So this could open maybe 70% of the space for microbiome research. So this is where I'm introducing my method, which is single cell phage amplicon barcoding sequencing, or single cell FabSeq, or single cell fabulous sequencing. Right here, I have the phages and bacteria that are being encapsulated in a PCR reaction mix. So this is a one-pot mixture where I could actually culture the droplets that have the PCR mix, allow the phages to replicate. So droplets that contain the right host with the right phage will actually be able to multiply. And then the droplets that do not have the correct host or phage will remain pretty neutral. And then you'll be able to barcode these by multiplex PCR and then Illumina sequence to demultiplex what the contents of the droplets are. So we have to come up with a bunch of different steps to basically validate whether this methodology is valid to actually study host-phage interactions. And the first one that I'm going to address is actually single cell encapsulation. If we're going to the single cell resolution, we have to ensure that we're actually capturing single virions per droplet. So when you're designing single cell experiments, such as 10x genomics or DropSeq, we know that the Poisson statistics is very relevant towards ensuring that we're actually capturing one particle per droplet. So right here, I have a plot showing the distribution of particles, or this could be phages or bacteria or cells if you're doing cells. And you could see that at a lambda value of 0.1, which is essentially the number of cells that are captured based on the concentration of cells loaded, that if we do a standard lambda of 0.1, 90% of the droplets will be empty and 10% of the droplets will contain at least one cell and the rest will be completely empty. You might not have any kind of doublets. If you expand upon that lambda range and basically load more different kinds of cells, you have a higher chance of developing doublets, as you can see from the red and from the blue and the green lines. So here I'm basically validating that I can go to the single cell resolution with phages. Here I have our standard T7 phage, which is a well-characterized kind of phage, loaded at a 0.1 lambda. So that means 90% of the droplets will be empty. And as you can see on the right, that 10% are actually fluorescing for GFP, which I used to stain the phages for DNA. So you can see that we are able to achieve single cell resolution right here. Next, we wanted to see whether we could actually replicate the biology inside of the droplets. So what I did was I took some phages at a concentration and I used this for bulk and droplets. And then I essentially allowed the phages to replicate within the bulk sample or the droplets, and then collected the phages and then titered them to make sure that phages were actually able to replicate in both conditions. You can see in this first plot right here, first and second bars, this is for T7 that are in bulk conditions, so basically milliliter spaces, and T7 droplets, which are in picoliter spaces. You can see that the titers are pretty similar after I collect all of the phages. And then after I let them replicate with their host, which is E. coli, you can see that the titer increases, indicating that the phages are able to replicate within picospaces. So next, we want to ensure if we're going to capture these phages and bacteria into droplets, we actually have to ensure that we can identify the contents of these droplets, and that involves DNA barcoding. So what we're going to do is actually barcode gene markers of interest. So this could be different kind of surface receptors on E. coli or the 16S to identify what kind of bacteria we're using, and then also studying different kind of structural markers such as the capsid or the tail fiber for phages. And then what we're actually going to do is each of the droplets will contain a unique barcode that has a P7 adapter. And then what you can do is actually stitch these barcodes together with whatever targeted loci of interest and be able to sequence them at the end after multiplex PCR. So since the barcodes are loaded at a limited dilution, that means 1 in 10 droplets will contain a barcode. 1 in 10 droplets will contain a template DNA. So if you do the math, 1 in 10, 1 in 10, 1 in 10, and 1 in 10, that means 1 in 1,000 droplets will contain a barcode, a phage, and a bacteria. But if you consider how many droplets are being generated with a microfluidic device, that means that there's essentially millions of reactions that can be collected. And what we hope to do by taking this data is we'll be able to tell whether there's a functional or non-functional host-phage interaction based on the abundance of barcoded reads that map back to the phage or map back to the bacteria. So right here, I have this little diagram showing a functional host-phage interaction where the phages were able to replicate and lyse their host. So you can see there's abundance of barcoded reads mapping back to the phage gene, and then a smaller fraction of them going towards the host. And then for non-functional, you'll see that the reads that are barcoded for both the phage and the bacteria are fairly equal. So over here, I'm going to show you a standard barnyard plot that's single cell RNA-seq. Folks might be able to recognize. Essentially, what I'm going to show is on this axis right here, you're going to see some green points indicating barcodes that map to phages only. And on this x-axis right here, you're going to see that there are going to be some red dots or barcodes mapping to bacteria, and everything in the middle that are blue plots are going to be barcodes that map to both phages and bacteria. And you could see right here that we can actually identify the contents of each droplet. So you could see that we could actually capture both E. coli and T7. So everything on the linear parts are just one sample alone, and then everything in the middle are the phages and the bacteria mixed together. So I basically showed you that we were able to complete 3 out of the 4 steps. And the real challenge came with step number 4, which is actually quantifying these interactions. Are we able to count how many phages we're able to replicate within the droplets? So this is just an overview, again, of what my method looked like. I encapsulated phages and bacteria in a PCR mix. I cultured them for the interactions, and then I essentially barcoded and sequenced them. So what I did was I took the same kind of loci of interest. I did the 16S for the host. I did a structural marker for the phage, and then I did the same barcoding schemata right here, where each droplet will contain basically 1 in 10. We'll have a barcode, and the rest will have some template. And then we should be able to see this trend where there is an abundance of phage genes if there's a functional host-phage interaction. But when you see this barnyard plot right here of a pre-infection sample that I did before the phage was able to infect the E. coli and a post-infection sample here on the right, of phages that were supposed to infect E. coli, you could see that there's no abundance of reads going upwards, indicating that the phages were not able to either replicate in the droplets or we weren't able to capture that with sequencing. So what we did next is that we actually went back. Maybe we stepped a little bit too far. We were a little bit too ambitious with going into sequencing right away. So what we did is I basically did the same experiment where I took titers of phages inside the PCR mixed droplets, and I titered T7 only and then T7 with a phage. So hopefully they replicated. And you can see as the bar is pretty much even, these phages were not able to replicate in droplets that contain the ddPCR mix. So we did 3 out of the 4 steps, but we're stuck on step number 4. What can we do? Well, essentially what we'll have to do is we're going to have to split it up. We're going to have to culture the phage-host interactions and somehow be able to combine it with the barcoding schemata. So that would require a different kind of technology platform that our simple droplet maker just cannot handle. So this is where I made a version 2.0, so a single cell FAB-seq version 2.0. So as you know, as we go on, different versions emerge from optimizing. So right here, what I'm going to do is actually encapsulate the phages and bacteria, not in the PCR mix for a 1-pot reaction, but actually in LB, which is their ideal conditions, incubate them to culture these different interactions. And what I'm going to do is actually inject them into another microfluidic device called a droplet merger, where I'm going to put them in a microfluidic device and essentially combine them together with the PCR mix that has the barcodes. And that way I'll be able to amplify the barcodes and Illumina sequence at the end. So this right here is an example of what my device is going to look like. So I spent some time like designing and optimizing and ensuring that structurally it does work for the experiment. So what I have right here is a very complex droplet merger, but when I explain to you, it's going to actually make a lot of sense. Right here, I have our Stanley droplet makers that are going to happen right here. So this is where I'm going to inject the PCR droplets, and here where I'm going to inject the phage-host interaction droplets, and they'll come together in this merging chamber right here where electrode or voltage will be applied so that the surfaces of these 2 droplets will destabilize, and as they move away from the electrode, they'll merge back together. And then I'll be able to collect them here. This saline reservoir right here is where I'm actually using it as a ground, so that way that no electrical current is going anywhere outside the chip. So I have the electrode and the merging chambers indicated right here in yellow and pink. So this shows what the merging chambers are supposed to look like. They're actually, you can see that the space is really tight for where the droplets come down, and that's to ensure that the droplets are able to stack on top of each other, so you get 1 and 1. And then as you go towards where the voltage is applied, the droplets will actually fall apart, and then as they move further down here, they'll actually be able to merge together. So here is a little video showing the droplets coming down. So you'll see that there's a large droplet and a small droplet. They're not being merged right now, but this ensures that I actually can get proper stacking so that one of each droplet can be put together to prevent any kind of doublets. So you don't have double barcodes or you don't have double templates. And right here is a video of what it looks like when I actually apply voltage. So on the left side here, you can see that droplets are coming from the left here, and then there's some reagents being injected on this middle channel right here. And you can see that the droplets are not combining, as you see like towards the right. Versus in this right-hand panel, you could see that with the voltage applied, you see that the droplets are actually combining together, and you could see that they are staying together as they move further away from the electrode. So in conclusion, and as a summary, I showed you basically the complexity of this project, about how there are so many different kinds of hills and valleys that I had to maneuver, and how I got 3 out of the 4 steps to go on. I'm hoping for the next colloquium, I'll have some updates to show you what exactly single cell FAB-seq version 2.0 will look like. So the future next steps is actually to combine the biology and the sequencing together. So what we're going to do is actually sequence these different host-phage interactions in parallel with known phages that have interactions with known hosts to actually validate that this works. And then we're actually going to scale up, since the Raman Lab does a lot of high throughput, makes a lot of high throughput libraries. What we're going to try to do is try to create high throughput operations together to kind of challenge the multiplex capabilities of this tool. And what we want to do is actually use this for the microbiome space, where we're able to actually screen and edit the microbiome with our engineered phage libraries and be able to tell certain information that has never been able to be told before. So I also want to address the RCR topic that the NIH standards wants us to do for our program. So what I'm trying to address is safe and respectful research environments, particularly towards people with visible and invisible disabilities. So this research was done under a supportive and understanding environment between the PI and the students. And then there was a lot of flexibility in terms of scheduling and for doing experiments as well. We also ensured that there was a recognition for every individual's unique needs. So not one disability looks the same as the other. And also, because we're in grad school, we might not think that we should go to medical appointments in the middle of the day, but sometimes you kind of have to. So students were able to take medical appointments during the day, and all research opportunities were presented in an equitable manner where certain opportunities were not taken away from people with disabilities. So yeah, I'd like to acknowledge my committee, Nicole, who's not here, David Schwartz, and Phil Romero and Ophelia Venturelli, who went on to Duke, but they passed on their blessing, which is their nanofabrication material to me as they moved. Also to Freeman Land at UToronto, who is my honorary mentor. And then the rest of the Raman Lab, who's been incredibly supportive during this time. Thank you. Yes.
I also found microfluidics very cool. I had a question. You had that song distribution of expected count sizes in each.
Yeah.
Did you ever consider expanding it so that you could? I mean, I really expect you would never have the power to get anything out of this, but if you considered looking at the data for multiple cells and multiple phage interactions, potentially that data would be analyzable just as a byproduct.
Yeah,
yeah. Remove it.
Yeah. So let me repeat that question for everybody on Zoom. Essentially, it was asked whether we could scale up the lambda value for the Poisson distribution so you could actually get more than one cell and then be able to basically analyze the contents of that cell and be able to pull some information from that. And for that question, I say yes. What we're actually planning on doing is applying some machine learning models. So if we have A phage and B phage with C host, then we see a different pattern. We could basically narrow down which was the common factor right there and be able to tell it's this host or this phage that was able to create a productive post-phage interaction. Yes.
I was just wondering how long they're together for, like the phage and the host, before you sequence them. So I was just wondering if, I know you want the phage to replicate, but is it also lysing the host at some point?
Yeah, so in oil droplets, they're actually stable for 24 hours. So I could culture them as long as 24 hours. And then if I wanted to do any more for additional lysis time, for example, if it takes like multiple days, then I would switch to a hydrogel, which I addressed before.
Oh.
Yeah. Yes.
This related to the previous question. So, which stage of phage infection would you expect to be captured from this sequencing technique?
Which stage would to be
Phage
captured?
infection.
That
depends on how long the.
Yeah, so that depends on the lysis. So what I'm specifically studying are lytic phages, so phages that are able to lyse and replicate within the host. So sometimes it ranges between what kind of phage and what kind of host. Sometimes it could take 45 minutes for a phage to replicate and create enough progeny. Sometimes it could take a couple of hours. So it's all really dependent on lysis times.
So it's specific to this type of phage.
Yeah, so it's specific to certain types of phages and hosts. And what we're hoping is, because microfluidic droplets are very tunable, if we could culture them for a long time, we could actually ensure that there's no bias, because the phages are contained within each droplet. We don't have to worry about them slipping out and then infecting other kinds of hosts. So we're actually ensuring that each of them are confined in their own space and they're able to do naturally what they're able to do in bulk reactions. Yeah, thank you. Yes.
Immediate follow-up on that. Have you considered taking a time sequence then?
Yes, so we actually were considering time sequences, or like a time lapse, a time course experiment too. Because I was only able to go to steps 1 to 3. I wasn't able to capture the lysis and the sequencing at the end. So I'm still working on that portion right now, but that is to be planned.